Good afternoon, everybody. I'm Francesco Celli, and I'm the division chair for endocrinology, diabetes, and metabolism, and endowed professor with the William Blackard professorship in medicine. And today is our honor to present the endowed Blackard lecture. Uh, and usually we invite distinguished scientists in the field of endocrinology and diabetes. And today is a particular honor for me. Probably some of you have heard us chatting before the talk. Well, our guest today is Dr. Alan Shuldiner, who's a truly world-renowned physician scientist. And most important to me, he was and he is my mentor. And I learned pretty much everything I do and I know and the way I approach medicine and in research from him. So this is real honor. I had the opportunity of seeing Dr. Shuldiner pipetting, and he's pretty good at. And probably the major learning point I got working with him is that if there is a research question that is worth pursuing, well, just go for it. And if there is not yet a technical tool to address this question, well, just work it out and invent the tool. And that's how I spent most of my fellowship, and I really truly enjoyed it. 3 seconds of bio. Dr. Shuldiner is a graduate from Lafayette College, went to med school at a small university in Boston called Harvard. And if I remember correctly, you did your internship and residency at Columbia, and then he moved at the NIH, where he completed his training in endocrinology, was in staff there, and then he moved to Hopkins and eventually to University of Maryland, where he worked first as division chair of endocrinology and then moved to associate dean for research. He now holds a part-time professorship at Maryland and is vice president at Regeneron for the Institute of Genetic Centers. And Dr. Shuldiner has been at the forefront of personalized medicine, starting on candidate gene approach, and then moved on genome-wide association studies. And his interest spans from diabetes, obesity, insulin resistance, dyslipidemia, cardiovascular disease, osteoporosis, and effectiveness of anticoagulation. So just a bit, and he published more than 400 papers. So I guess I talk way too much. Alan, go.
Well, thank you so much, Francesco. It's really an absolute pleasure and honor to be here virtually to deliver the William Blackard lecture. And as Francesco mentioned, he is probably the most prolific of my mentees, and it's always fantastic to see all that he's accomplished in his career and how great he's doing at VCM, and ultimately I'm sure will eclipse any of the accomplishments that his mentor has made over the years. So can you all see my screen?
Yes.
Okay, wonderful. Okay, so I know that I need to provide a disclosure. Actually, I have 2 full-time jobs. One of them is at the Regeneron Genetics Center, where I'm vice president. The second is that I'm continued as the John Whitehurst Professor of Medicine, now part-time at the University of Maryland School of Medicine. So I have to say I've never met William Blackard, but certainly I know his work very well. Both Francesco and I began some of our training at the NIH. My mentor was Jesse Roth, and Jesse had exactly the same interest as William Blackard did about the same time, and that is during the 1960s and beyond, insulin sort of just being understood was to figure out how insulin works and what causes insulin resistance in patients with type 2 diabetes and obesity. And looking back at some of Dr. Blackard's work over the years, really seminal work that you only know is seminal by looking back at it. And the thing that impressed me really quite a lot is that Dr. Blackard, through really his entire career, 40 years of publications, as you can see here, well over 100 publications in PubMed, was hyper-focused on that question. How does insulin work, and how is its inaction relevant to health and disease? And I'm showing you here, literally from PubMed, his first publication in 1962 and his last publication in 2000, essentially focused on the same question. And if you don't have time to read all of his 124 plus publications to look back, I'd point you towards the MCV Quarterly in 1977, when he wrote just a wonderful overview of what was known at the time about insulin receptors. Fast forwarding to what we know now, it is really historically a very interesting article that I would point you all to. So in terms of today's lecture, what I'd like to do is focus on precision medicine. And of course, with my interest in endocrinology and diabetes, the focus will be more on discoveries in genomics that allow us now to be thinking about more precision medicine approaches for endocrine disorders like type 2 diabetes, and also metabolic complications of obesity, particularly non-alcoholic fatty liver disease. So what I'm going to do today is describe how human genetics is being applied to therapeutic development for therapeutic target discovery, but also in clinical trials to begin to stratify patient populations for increased efficacy. I'm going to describe how founder populations, I work very closely with the old order Amish now over more than 20 years, how these founder populations can provide special opportunities for novel gene discovery and ultimately insights into human biology relevant to all humans, not just those that are parts of these founder populations. And finally, fast forward a little bit to describe how exome and whole genome sequencing can be applied to improve human health and disease in healthcare systems and even across populations, so-called population genomic health. So when we talk about therapeutic development, one of the things that I've learned the last 6 years being in industry is that making new therapeutic agents is really hard. And despite a really marked increase in investment in research and development in pharmaceutical companies and in the biotech industry, the number of new molecules and entities that are actually entering into FDA approval is not increasing. And this is because more than 90% of molecules that actually enter phase 1 clinical trials fail to demonstrate sufficient safety and efficacy to gain regulatory approval. And recall, these phase 1 clinical trials are actually at a point where probably hundreds of millions of dollars have already been invested in that molecule, only for naught in terms of having to put that molecule up on a shelf and not develop it further. And it turns out that most of these clinical trials failed because of decreased efficacy, or lack of efficacy, I should say, and or unanticipated toxicity. This has led the field to the conclusion that the current business model for developing these therapeutic agents just doesn't work. And the preclinical models that we use, whether they be mice or even non-human primates, are just poor predictors of clinical benefit in humans. And this has now led us to the idea that compounds that are supported by human genetic evidence are substantially more likely to succeed in terms of efficacy and decreased toxicity. So now, with all we know about the human genome, in fact, we know more about the human genome than any genome of any species, we're now beginning to think about applying human genetics for more efficient therapeutic development. And now there are, several, terrific stories of how human genetics have driven novel gene discovery and ultimately new therapies. I'm showing you two of them here. One of them that many of you know about is the Pcsk9 story. The initial discoveries were actually made in rare families that had gain of function mutations in Pcsk9. And these individuals had a new form of familial hypercholesterolemia. Not long after that, population studies identified individuals that actually had loss of function, LOF variants in Pcsk9. And these individuals actually had very low LDL cholesterol levels and had marked protection from coronary heart disease. This obviously led to the idea that if we could mimic an antagonist of Pcsk9 as a therapeutic, this would have great efficacy in terms of lowering LDL cholesterol levels and protecting from heart disease. In addition, these individuals who had these loss of function variants from birth didn't seem to have any untoward side effects of inhibition of Pcsk9, suggesting that genetically this would be not only an effective therapeutic target, but also a safe therapeutic target. And now, as you know, there are at least two FDA-approved medications on the market, antibodies that inhibit Pcsk9. They're quite effective in lowering cholesterol, especially in patients with familial hypercholesterolemia, and ongoing clinical trials also showing cardioprotection. The second example that I'll show you is one that I'm particularly proud of because the initial discovery back in 2008 was made in my lab, in which we discovered in the Amish the first null mutations in ApoC3. And these individuals had very low triglyceride levels and also had cardioprotection. Two population studies that actually confirmed these findings in the general population, and now several pharma and biotech companies developing ApoC3 inhibitors for the treatment of hypertriglyceridemia, potentially also for the treatment of coronary artery disease. And there are now multiple other examples that are emerging from human genetics to novel gene discovery to new therapeutic targets. So now, in terms of therapeutic development in the pharma and the biotech industry, we've really turned this paradigm completely on its side. And we're now beginning with humans and human genetics to essentially identify new drug targets. We're using them to identify new indications for drug targets and programs. We're using human genetics to de-risk a particular therapeutic target. And of course, we're also using human genetics for biomarker discovery in the pharmacogenetics arena. And it's only until we have human genetic evidence that a particular drug target will be safe and effective that we then begin to use the tried and true conventional preclinical models to dive more deeply into the biology and the underlying mechanisms. So as you can see, starting now with humans, going to the preclinical models, exactly the opposite of how business was done before the advent of human genetics. And at the Regeneron Genetics Center now over the last 6 years, we've been working with academic collaborators from around the world to sequence patient populations to identify these genotype-phenotype associations that ultimately identify new biology and potential new therapeutic targets. We now have over 100 collaborations from all over the world with academic collaborators and healthcare systems. We've sequenced over 1.5 million exomes from patients from all over the world. And our model essentially is that this is all being done in the pre-competitive space. We encourage publication and getting all of this data out there. I have to say, in the few years that I've been in industry, I've been publishing more than I ever have, and even in higher impact journals, since these exome sequencing projects are at a scale that is totally unprecedented in the field. So let me tell you about one such collaboration. It's a collaboration we have with the Geisinger Health System. The Geisinger Health System is located in central Pennsylvania, and they're essentially an integrated healthcare system similar to the HMOs like Kaiser Permanente, for example. They care for 1.6 million participants, predominantly European Caucasian, in this central Pennsylvania area. They were very early adopters of the electronic health record, and they have very nice longitudinal records of a very stable patient population since 1996. On average, patients have at least 18 outpatient visits over a period of many years, and this has really been a very rich database for genetic discovery. Recruitment is ongoing, and to date, over 200,000 Geisinger patients have been consented into the MyCode Discover biorepository cohort. At the Regeneron Genetics Center, we've sequenced over 175,000 of these individuals, and we're now uniting the exome sequence data with the rich de-identified electronic health record data to make many incredible discoveries that have been leading to new drug targets and new biology. So the one that I'm going to tell you more about is this story, a story in which first I'll tell you about what we've found in sequencing 50,000 patients from an integrated healthcare system, and then dive a little bit deeper into the story of a new target for non-alcoholic fatty liver disease. So 50,000 patients have been sequenced, and you can see that across the entire Geisinger Health System, we have very rich electronic health record data to unite that exome sequence data in. The upper part of the slide shows you that for example, over 500,000 Geisinger patients, we have 10 years or more of longitudinal electronic health record data to mine. And you can see then when you look at the most prevalent diagnoses and the most prevalent laboratory values, we have hundreds of thousands of individuals who have common diseases such as hypertension, hyperlipidemia, type 2 diabetes, and the like, hundreds of thousands of individuals in which we can potentially find new connections to genetics. And we have millions of laboratory values and millions of, to again, connect to human genetics. So this is the phenome, the longitudinal phenome that we work with. And then we can ask the question, what do we know about the genomes of these individuals? And this data shows you a summary of what we know about the first 50,000 discovered participants that were sequenced. You can see that in this exome sequencing project, we identified over 4 million variants. And the vast majority of those variants are rare. They have a frequency of 1% or less in the population. So this cues you into two things. One is that it's clear that to understand these rare variants, we're going to have to sequence hundreds of thousands, if not millions of people to identify enough individuals with these rare variants to make sense of them. And the second thing that it tells us is that the common variant GWAS analyses that have been done for the last 10 or more years are likely to be missing a lot of really interesting genetics that live in the more rare variant space. You can see of these 4 million or so variants, about 175,000 of them are variants that would be predicted to knock out the function of that protein product, so-called predicted loss of function variants, PLOFs, 200,000 of them in the 50,000 sequenced. And again, it's our challenge to find out what those loss of function variants do or are associated with vis-a-vis the electronic health record. If you boil this down to an individual person, and any of you in the audience is probably no different from a typical Geisinger patient, a typical individual is heterozygous for a predictive loss of function variant in about 21 of their 20,000 genes. And each of us is a homozygous predicted loss of function carrier, essentially a human knockout for one gene in the genome. So this is what makes each of us special in many ways. I think each of us, all of us, can consider ourselves human knockouts. If you go to the 50,000 exomes, in fact, we have 17,000 genes, 92% of the entire genome, in which we have at least one individual that's heterozygous for a predicted loss of function variant, and over 1,300 genes, 7% of the genome, that we have at least one individual that is essentially a knockout for that gene. And again, understanding these individuals' health through the electronic health record is what we really want to understand. So I'm going to give you one nice example of a new target for chronic liver disease, and in particular non-alcoholic fatty liver disease, disorder that with our obesity epidemic has become an epidemic in this country and in westernized countries all around the world. Now the leading cause of chronic liver disease, and one in which we really have no good targets to understand. So I'll walk you through the main data. Essentially what we performed was an exome-wide association study, an XWAS, of ALT and AST levels in these close to 50,000 Geisinger patients for which we have exome sequence data and also rich electronic health record data. And you can see that with these Manhattan plots, we have several hits that are showing genes and gene variants that are significantly associated with ALT levels on the top and AST levels on the bottom. One of the genes that was relatively unique was a gene called HSD17B13. And so we went ahead and looked into that gene in a bit more detail. And if you can see on the right-hand part of the slide, we looked essentially at chronic liver disease in these individuals. And not only were we able to look at the Geisinger Health Study, but also we had sequenced about 2,000 members from the Dallas Liver Study as well. Actually, more like 4,000 members from the Dallas Liver Study as well. And you can see when we extracted ICD-9 and 10 codes here, you can see that individuals with the variant, the heterozygotes and the homozygotes, were actually significantly protected from chronic liver disease. And it didn't really matter so much how we sliced the pie. We were protected from alcoholic liver disease, alcoholic cirrhosis, non-alcoholic liver disease and cirrhosis, and even were protected from hepatocellular carcinoma, which we know is one of the end stages of chronic liver disease. We were able to actually replicate this data very nicely in a completely independent data set, the Dallas Liver Study, and this really suggested to us that we have a new molecule, a new gene in which we could potentially target for the treatment of chronic liver disease, including non-alcoholic fatty liver disease and NASH. One of the very cool things about the Geisinger Health System is that they have a very large bariatric surgery program, and that bariatric surgery, they got liver biopsies in more than 2,000 of their morbidly obese patients. We have histology on those individuals, and we were able to essentially relate this HSD17B13 genotype to liver histology. And you can see in individuals who carried this variant, the TA variant, indeed, they didn't have, they, if anything, had more simple steatosis, essentially fatty liver, but you can see what they were actually protected from were the more advanced stages of fatty liver disease, so-called NASH and NASH fibrosis. Similar thing is shown on this slide when you compare simple steatosis patients to those who have normal livers. The variant didn't seem to relate much to simple steatosis, but did seem to protect from NASH and NASH with fibrosis. So this really suggests that this particular gene doesn't necessarily protect from fat in the liver, but it suggests a role in terms of protection from the progression of simple steatosis to more advanced chronic forms of liver disease. Again, an indication that this could be a very good therapeutic target to protect from advancing chronic liver disease, even in the setting of a fatty liver. So what is this gene? For one thing, the variant is an insertion, and it alters a splice site in the gene, and that results in a frame shift and an early chain termination of the HSD17B13 protein. So this is a predicted loss of function variant, and you can see then when we looked at RNA levels in these liver samples, those who were homozygous for the normal allele made plenty of mature HSD17B13 mRNA, and those who were homozygous for the variant made little or no mature HSD1713 RNA. On the contrary, they made much more of the aberrantly spliced mRNA, one that as I said, is a frame shift and an early chain termination of the protein. And in fact, when you look at the protein levels of HSD17B13 in those liver biopsy samples, homozygotes made little or no mature HSD17B13. And we were able to show that HSD17B13, a protein that really is very little is known about, actually localizes to the lipid droplets in HepG2 cells, again, suggesting some important biology for fatty liver and the progression of fatty liver to more malignant forms. So what is HSD17B13? It turns out that it's a member of the hydroxysteroid 17-beta dehydrogenase family, one that endocrinologists know well. This particular family is involved in the metabolism of sex steroids and fatty acids. The endogenous substrate for HSD17B13 is unknown, but certainly the genetic findings stimulated us to try to understand much more about what the endogenous substrate actually is. So we went ahead and tested 265 putative substrates and found that estradiol and some bioactive lipids were actually substrates for this HSD17B13 enzyme. You can see on the right hand of the slide enzymatic activity to estradiol and its conversion to 17-beta. And you can see an enzyme that essentially is an active one for that substrate. You can see in the lower part of the slide the fact that the normal protein is quite enzymatically active, but the protein that's produced because of that TA mutation is essentially an inactive enzyme, a truncated enzyme that is totally inactive. Same thing on this part of the slide. So individuals who are loss of function carriers of HSD17B13 are the ones that are protected from chronic liver disease, suggesting that if we could make a therapeutic agent that inhibits HSD17B13, this could be very effective for the treatment of non-alcoholic fatty liver disease. And this is where we are now. And of course, this work is published, and I'm sure that many other biotech and pharma companies are running after inhibitors for this new enzyme and new drug target. So this is an example of how we applied high throughput human genetics at the level of exome sequencing and a healthcare system to uncover new biology and a very promising new therapeutic target. Let me move now to a couple of other stories. As many of you know, much of my career has been spent studying the genetics of complex diseases in founder populations. Founder populations are very unique in terms of opportunities for novel gene discovery because by definition, these founder populations have much more genetic homogeneity. There's also the opportunity for genetic drift in which rare variants that get into the population over generations have the opportunity to increase in frequency. So these rare variants in the general population may be markedly enriched in these founder populations, making them much easier to discover. Many of these founder populations practice endogamy, and there's a lot of consanguinity and very large families. And of course, this provides these drifted variants the opportunity to occur in their homozygous state and to identify recessive alleles where you would find them literally nowhere else on the planet. And finally, founder populations tend to practice a much more homogeneous lifestyle, which introduces fewer confounding influences when you're looking for genetics. The old order Amish are a perfect founder population for novel gene discovery. And this was not my discovery, but that of Victor McCusick's back in the 1950s when he began working with the old order Amish to begin to catalog genetic disorders that occur in that population, even before the structure of DNA was known. They were founded by about 200 to 600 individuals. They migrated from Switzerland about 15 generations ago. They've grown to a population of more than 40,000. They've maintained themselves as a very isolated community in that they've maintained cultural traditions and also have very large sibships and a moderate amount of endogamy. So all of the characteristics that provide opportunities for novel gene discovery exist in the Amish. And now over more than 25 years, and Francesco has been involved in some of these studies as well, we've made some really cool findings in the Amish that most of which are generalizable to the general population. Bullet through them very quickly now. I told you already about the ApoC3 story. We also found out that the Amish are markedly enriched for familial hypercholesterolemia. In fact, 1 out of every 8 Amish carry the R3527Q ApoP variant. And hypercholesterolemia is a major health problem in the Amish, one that we discovered through genetics and are now applying to the community for early diagnosis and early treatment of severe hypercholesterolemia in that population. Colleen Damcott in our group characterized a very interesting loss of function mutation in hormone-sensitive lipase that has provided some very interesting new insights into the biology of lipid metabolism and its relationship to type 2 diabetes. Through exome sequencing, we've been working very closely with the Clinic for Special Children and have diagnosed through exome sequencing many new novel genes that cause severe undiagnosed diseases in Amish and Mennonite children. And a lot of these new discoveries have been applied now in the clinic, not just for diagnosis, but also for novel treatments. We also performed a study in the Amish where we treated them with clopidogrel, Plavix, and ultimately identified markers that are associated with clopidogrel response. And this is a story that I'm going to tell you very quickly in a few slides to follow. And finally, I'll tell you a quick story, a new one actually led by Liz Streeton in our group, showing that actually 1 out of 45 Amish carry a pathogenic mutation in KCNQ1. And these individuals are at increased risk for sudden cardiac death. We'll tell you a little bit about how we've begun to implement genomic medicine in the community to prevent sudden cardiac death in large numbers of Amish in the population. So the first very quick story is a story of pharmacogenetics, where we're trying to identify genetic markers that will predict responsiveness to drugs so that we can better target individuals and medications for those individuals to optimize efficacy and to minimize adverse side effects. And this story is one of clopidogrel or Plavix. For this audience, I don't need to remind you of its anti-platelet actions and its use in coronary artery disease. And although it's used very widely, there is great inter-individual variability in response to clopidogrel. And depending upon how you define response, anywhere between 4% and 1/3 of individuals can be considered resistant to clopidogrel. So we began a study that we call the Amish Pharmacogenetics of Anti-Platelet Intervention Study, the PAPI study. We took 668 healthy Amish individuals and we treated them with clopidogrel for 1 week. We measured platelet aggregation before and after therapy, and we show the results on the right-hand part of the slide. So you're looking here at platelet aggregation, upper panel before clopidogrel, lower panel after 7 days of clopidogrel. You can see that on average, the population responds to clopidogrel. There's a shift in terms of antiplatelet activity in the population. But if you look at specific individuals in that population, there are some that have very good responses to clopidogrel and others that start in the same place but have virtually no response to clopidogrel. And so the population responds to clopidogrel. This is what the FDA tends to look at when they approve a particular drug, but indeed there's great individual variation in response. And we were able to go ahead and look at heritability of clopidogrel response just to ask the question whether relatedness, forget about performing a single genetic test, just whether relatedness could explain response to clopidogrel. And as it turns out, 70% of the variation in clopidogrel response could be attributed to just by relatedness, suggesting an important genetic component. We went ahead and did a genome-wide association study for clopidogrel response. You can see we identified on the upper left-hand panel a screaming hit for clopidogrel response. It turns out when we mapped this down, it turned out to be a loss of function variant, actually a known one, in one of the cytochrome P450 genes, CYP2C19. You can see when we genotype for this STAR2 variant, in the absence of clopidogrel, it didn't predict platelet aggregation at all, but in the presence of clopidogrel, predicted platelet aggregation quite remarkably. And those with one loss of function variant and those with two loss of function variants were shown to have a much poorer response to clopidogrel, less aggregation, less response, or I should say more residual aggregation in the presence of clopidogrel. And when we took that discovery to a general coronary artery disease population in Baltimore, the Sinai Hospital cardiac cath lab, we were able to show essentially that those who had a stent placed, who were placed on clopidogrel and retrospectively genotyped, that those who had one or more copies of this STAR2 allele had more coronary artery events, about a 2.4 increase in risk for coronary artery events while on clopidogrel, suggesting indeed that these are poor responders and potentially these are individuals that should get an alternative antiplatelet therapy. So it turns out this STAR2 variant is quite a common variant, and a third to a half of individuals carry at least one copy of this STAR2 loss of function allele. It accounts for about 12% of the variation in clopidogrel at the level of platelet aggregation, and as you've seen, is associated with about a 2.5-fold increased risk of a recurrent cardiovascular event, an effect size that I think most would believe is quite clinically relevant. We now know how this works. It turns out that clopidogrel is a prodrug. It's only its active metabolite that ultimately binds irreversibly to P2RY12 receptors on the surface of platelets to inactivate platelet aggregation. And it turns out that CYP2C19 is one of the important enzymes responsible for activating clopidogrel. And of course, if you have loss of function variants in CYP2C19, you don't make the active metabolite effectively. And this is the reason why these individuals don't respond as well in terms of inhibition of platelet function. Right around the time that we published this data, other reports came out as well. And the FDA took note and back in 2010 mandated this boxed warning on clopidogrel. It was updated in 2019. And essentially, this is making clinicians aware of the fact that there are CYP2C19 poor metabolizers and that these individuals will be less responsive to clopidogrel and that physicians should consider use of another antiplatelet agent in these individuals. And as it turns out, around the same time, two alternatives for antiplatelet therapy became available, prasugrel and also ticagrelor. And at the same time, almost as well, Plavix went generic. So this really set up a perfect scenario for pharmacogenetics of antiplatelet therapy. The idea being that 2/3 of individuals that were good metabolizers of clopidogrel should get clopidogrel, the inexpensive drug, and we should reserve the more expensive, the newer antiplatelet agents for those who are intermediate or poor metabolizers. And in addition, we would spare that increased risk of bleeding that's present with the newer agents in a half to 2/3 of the population by continuing them on clopidogrel. So seems like a pretty clean story, but even now, 10 years after the publication of those papers, 10 years after a boxed warning on the label of Plavix, most cardiologists are still not performing genetic testing to personalized antiplatelet therapy. This slide shows you when you poll a bunch of cardiologists, why aren't you doing genetic testing in the cath lab? A lot of them will quote the fact that there are no prospective randomized clinical trials showing that clopidogrel matters, or that I should say genotyping for CYP2C19 matters. Just in the last year, that's changed. There are actually 2 published prospective randomized clinical trials that I'll show you shortly. Healthcare and provider education is a big deal. Many cardiologists, particularly in the community, have never heard of CYP2C19, and the fact that a genetic test is actually available. Reimbursement for a long time has been an issue, but now with the randomized clinical trials, many third parties are reimbursing for genetic testing for CYP2C19. And despite all these barriers and the fact that this is not really caught on yet in a widespread way, patients really understand precision medicine. They know very well that some of their family members respond to certain drugs and others don't. And they really want to get this genetic testing and get more precision therapies. Well, as I mentioned now, there are 2 randomized clinical trials that have been performed just in the last year and a half. Both of them really point towards efficacy and clinical utility of genetic testing for precision antiplatelet therapy. This one statistically significantly showing non-inferiority of a genotype-guided approach, and also the fact that there's significantly less bleeding in when using a genotype-guided approach. And the second one, one in which trends in the same direction were observed, a study that was designed a little bit differently than a non-inferiority trial, but also showing about a 1/3 reduction in coronary artery events in people who get genotype-guided therapy. This was not a statistically significant result, so it depends upon how you want to interpret this study, cup half full versus cup half empty, but it did show at least a numerically significant decrease in hazard ratio using a genotype-directed approach. So I could tell you at the University of Maryland, we really believe in precision medicine. We really believe in precision antiplatelet therapy. And over the course of time now, we have figured out ways in which we can do CYP2C19 genetic testing for all patients that come through our cardiac catheterization lab. We have a test in which we have a 4 to 5 hour turnaround time. And so from the time the patient gets his coronary catheterization to the time that they're discharged, they have a CYP2C19 genotype in their chart. And our cardiologists can use that information accordingly to optimize antiplatelet therapy in those patients. The last story that I'm going to tell you very quickly is that about long QT syndrome in the old order Amish. And again, I don't need to remind this audience about long QT syndrome and sudden cardiac death. Pretty rare in the general population. And it's due to mutations in several genes, but the predominant gene is KCNQ1, a potassium channel expressed in the heart. And one of the key things about long QT syndrome and these mutations is that this is a treatable disease in that an inexpensive beta blocker and also avoiding provocations that might cause arrhythmias in these individuals are very effective at preventing sudden cardiac death in these patients. So with exome sequencing in the old daughter Amish, we actually identified a missense mutation in KCNQ1, threonine at codon position 224, substituted for methionine, very rare in the general population, but it turns out that it has a frequency of a bit over 1%, a minor allele frequency about 1% in the Amish, which translates to the fact that 1 out of 45 Amish carry this mutation and have long QT syndrome. In ClinVar, this was characterized as a variant of unknown significance, but with much work that was done by my colleague Liz Streeton, we were able to actually show that this particular variant causes a decrease in function of this potassium channel. It's highly associated with QT interval, and in going back to these individuals and asking them about syncope and sudden death, you can see that they, as well as family members, have a marked increase in the frequency of syncope and sudden death. And in fact, when we went back to these families, so we were able to reclassify this as a variant from a variant of unknown significance to pathogenic. And then of course, in characterizing it as pathogenic, we felt an obligation to return this results to Amish individuals and to provide them with therapeutic recommendations, hopefully to prevent sudden cardiac death in a proportion of those 1 out of 45 individuals in the Amish who have this mutation. When we went back to these families, really tragic stories, and these are examples of 3 families and their family histories of sudden death. You can see these families had a lot of crib deaths, they had a lot of stillborns, a lot of sudden death in in children and adolescents. Classic stories of children jumping into ponds in May where the water was cold, and despite being very good swimmers, having drowned. Another child walking to school on one winter morning with sudden cardiac death. All classical stories of KCNQ1 mutations, long QT syndrome, and sudden cardiac death. And we've now been able to implement genetic testing in the community, and we believe this will have important implications for preventing sudden cardiac death in the community. So in the last 5 minutes, let me now zoom out. You've heard a lot about how we're sequencing a lot of exomes in many populations, making new genetic discoveries that are translating to new therapeutic targets. You heard a story about how we've been able to make one discovery, actually a couple of discoveries if you include the ApoB story, in a special population like the Amish that has implications for population genomic health. How does this apply to the general patient population? And this is where I'm going to give you a glimpse of the future, the idea and the question that we'll pose: what if every patient's genome, including every patient at VCM, were actually sequenced? How can we use that information? What would we know? And this experiment has already been done. It's been done at the Geisinger Health System. I've already explained to you that we've sequenced over 170,000 patients at the Geisinger Health System. Of course, the Regeneron Genetics Center is using that information to identify new biology and new drug targets. But at Geisinger, they're using this information to actually return actionable results to patients for patient care. And I won't walk you through this whole slide, but the bottom line is that Geisinger has identified 76 genes for which, if there is an individual that has a mutation in one of those genes, it is causative of 27 different diseases that are medically actionable. And these are diseases like hereditary breast and ovarian cancer, Lynch syndrome for colon cancer, familial hypercholesterolemia, hypertrophic cardiomyopathy, and multiple more. And so now they can use a genotype first approach to be very proactive about identifying people that have these mutations that maybe have not yet fully clinically manifested themselves and for which an action could actually prevent or delay their disease. It turns out that about 3.5% of study participants actually will test positive for an actual variant. So this has implications for health systems. If you think about how they're going to have to grow their genetic counseling programs, if they were to sequence every one of their patients, you can estimate that about 3% of them are going to need to be followed up in a genetics clinic by a genetic counselor. And of course, all patients have clinically relevant pharmacogenetic results, 100% of them. And this paper published not long ago by the Geisinger group goes through their experience to date. You can see all of the genes and the mutations that they've identified that cause all of these actionable diseases that they have been able to return results back to patients for better treatment and prevention. At the end of the day, they've, at least at the time of this paper, have reported results back to over 500 patients spanning 21 different disorders and 40 gene mutations that, 40 genes and mutations in 40 different genes that cause these 21 diseases. But you know, it doesn't get real and it doesn't get precision and personal until you begin to listen to individual people's stories about how the sequencing of their genome have made remarkable differences for their health and the health of their families. So this data is all on the web. If you access the Geisinger website, no issues here with HIPAA. Barbara Barnes is a 57-year-old grandmother. She was bringing up 3 grandchildren at the time. She entered into the MyCode study. She was found to have a pathogenic mutation in the BRCA1 gene. With genetic counseling, she was given that information and her response was, okay, so what do I do next? I have 15 more years to go until my grandchildren are raised. She had genetic counseling. She was found to have a workup. She had a negative mammogram. She elected to have a bilateral salpingo oophorectomy. At surgery, she was actually found to have a stage 1 cancer in one of her fallopian tubes. My gynecology colleagues will tell me that the natural history for Barbara would have been to present 5 or 8 years later with unresectable stage 4 ovarian cancer. So having this molecular diagnosis at age 57 allowed a very treatable form of her cancer to be diagnosed. She had chemotherapy and is expected to have an excellent outcome. And of course, now her daughter is being tested for the BRCA gene as well. And finally, Kim Mermert, 66-year-old Geisinger employee. She tested positive for Lynch syndrome and obtained genetic counseling. And for her, she's going to get more frequent colonoscopies. And 2 of her children will begin to be screened for colon cancer at an earlier age than might normally be recommended. So to conclude, I'm happy to tell you about amazing progress that has occurred in our advances in understanding the human genome and how this new information is being used for therapeutic development and also for the implementation of precision medicine in special populations like the Amish, but that last few slides showing you a glimpse of the future, how it will be applied to the general population. These genetic experiments of nature have really been very informative. And maybe my last bullet point and point that I'd like to make is that these partnerships between industry, academia, and healthcare systems is where it's at in terms of accelerating genomic discovery, ultimately translation of these discoveries to the implementation of precision medicine. So with that, I'll conclude. Obviously, huge numbers of people who have been working on this project and are most deserving of acknowledgement. Large center program at the Regeneron Genetics Center and collaborators literally from around the world. In this case, I've told you about the work at Geisinger, the University of Maryland, and also functional studies for KCNQ1 that were performed at Vanderbilt. thank you so much for your attention. I'm happy to answer any questions.
Thank you, Alan. That was a spectacular lecture, and it encompassed all internal medicine, and that's fantastic. I'd like to share a question from Dr. Mayer, and in essence, it refers to the question of I'm just quoting, knowledge of genetic relevance of medication efficacy is likely to be critical for health. But what are your thoughts on the ethical questions around genomic testing information having bearing on blood relatives who may not have been informed about the consequences of having information and around privacy concerns?
Yes, so I apologize for skirting around or not even mentioning the ethical issues, but it's not to say that I acknowledge that these are really important issues that we as a society need to deal with. And my quick opinion here is that no one size fits all. I think that at the end of the day, each individual, each patient, needs to decide for themselves whether they want to know anything about their genome, and if they do, who to share that information with. I think at the end of the day, everyone will have a hard drive or a thumb drive, with their 3 billion base pair genome sequence with variable interpretations, and they will decide who and what they wish to share with. The question about those then implications of individuals' decisions and how those individual decisions have implications for family members is a really tough one. And again, one size doesn't fit all here. I think families are going to have to get together, and figure out how they want to function as a unit as more and more of their family members make decisions about wanting their genomic information.
I'd like to ask you one question, looking at the other side. So as practicing physicians, we see quite often the very interesting presentation. So the outlier in response to one drug, very odd clinical presentation, which brings near and dear to me the candidate gene approach. But how do you think these observations can be fed in a larger picture, and again using the experiment of nature and moving back to the research and discovery phase?
Yes, thanks, Francesco, for that question. So, I wasn't able to go through the study designs for the 100+ collaborations that we're working on at the Regeneron Genetics Center. I told you about the very large unselected patient populations and gave you some examples there, but fully agree. And we have many studies underway in which we are indeed recruiting individuals, or I should say our collaborators are recruiting individuals who lie at the extremes of one phenotype or another. And you're absolutely right. These are individuals in which you essentially have stacked the deck for the likelihood of finding something new and interesting from a genetics perspective. And we have been doing a lot of sequencing at the extremes of phenotypes. One that will ring near and dear to you, Francesco, since both of us started in the geriatrics and the aging arena, is that we're very interested in sequencing the genomes of super centenarians, individuals who have lived to 110 and beyond. Very few of them out there, but boy, their genomes should teach us something. And with regard to candidate genes, I won't say that candidate gene approaches are dead, but I will say that it's cheaper to sequence an exome than it is to Sanger sequence a single gene these days. So the candidate gene approach has morphed a bit. And I think most of us would agree that we would do an exome sequence, but maybe in advance, we would put together a small list of candidate genes in which we would go in to analyze the exome in more of a hypothesis-directed way than an exome-wide hypothesis-free way.
Got it. Okay. And one question from Dr. Alan Dow, and the question is, do you see any role in looking big genomic data to environmental factors and to some degree response to social determinant of health?
Yeah, I love that question. So in many ways, if you think about it, those are the kinds of genes you really want to know about, because you know that there's something in the environment, some social or other intervention that you can target those individuals for that will actually have utility. So I think the holy grail really is to understand gene-environment interactions. And it is sort of a holy grail in many ways because it is just so hard to measure the environment over time, since these chronic exposures, occur over the course of an individual's lifetime. There's a lot of interest by NIH and other groups to figure out ways in which we can use mobile technologies and other technologies to better measure the environment so that we can then apply the powerful genetics to actually identify those gene-environment interactions. So a very active area, one that I think we need to really spend more time doing. And then of course, there's the interaction of the environment with the epigenome, alterations in the germline genome that occur through DNA methylation and the like. And this is yet another area that's going to be really important to dive more deeply into over the years to come.
That's great. I am conscious of your time. By the way, this is, in a way, it's a down payment. On purpose, I did not ask to have individual meetings at this time because Dr. Shuldiner, sometime once this little virus is gone, will come and visit VCU, and is going to spend some time with us and with the food scene in Richmond. So once again, thank you for this spectacular talk, and looking forward to see you again in person. Bye, Alan. Thanks.
Thank you, Francesco. Take care.
Bye-bye.
Bye.